In this video, we'll talk about another very important topic of this chapter, that is population genetics and Hardy-Weinberg's law or principle. Law or principle. Now let us understand these two things first, what exactly they mean, the terms, and then we will take up some more important things about the principle. Population genetics means the study of the frequency of genes in a population. That is, we are studying the gene frequency in a population. That is known as population genetics. Now, Hardy-Weinberg Principle or law says that the relative frequencies of alleles remain constant from one generation to the next, or from generation to generation. From generation to generation in a population where the organisms are sexually reproducing. In a population where organisms are sexually reproducing. This is the law or the principle. That means in a population, the relative frequency of alleles remains constant generation after generation. The condition is that the organisms in that population should be able to breed sexually, and this is applicable when number one, the population is big. That means number of individuals in that particular population is much higher. Then in that case, this principle is going to hold true. Second, in this population, the organisms show random mating. Third point or condition when this principle would be applicable is that there are no mutations in those individuals, and if mutations take place, they are in both the directions. So no mutations. If mutations take place, they are in both directions. And the fourth condition when this law will be applicable is that all the organisms which are born, they survive to their reproductive age. So all organisms survive and all of them have pretty much uniform reproductive potential and have uniform or same reproductive potential. If these conditions are fulfilled, then Hardy-Weinberg law or principle holds correct. And the law says that if these things are fulfilled, in a given population, the relative frequencies of an allele, that is allele of a gene, would remain constant. Conditions are these: the population has to be big, they should show random mating, no mutation, and if at all mutations are taking place, they are in both the directions. All organisms survive and they have equal reproductive potential. Now, to understand this law even better, we will talk about the expression of these alleles, and we will come to that equation also. So let us talk about what is the dominant represented by or the recessive one. Let us see the expression of these alleles. The dominant allele, if we are using say A to represent a particular gene, the dominant will be represented by capital A, and here in the principle it will be represented by P. So if the condition is homozygous dominant, it is p², and small a would be represented by Q, that is the recessive allele we are talking of, and homozygous recessive condition is represented by q². There is one more condition which exists, that is heterozygous condition, that is capital and small a. It is represented by 2pq. So according to Hardy-Weinberg, in a population, homozygous dominant, homozygous recessive and heterozygous make the gene pool. And if all the conditions that the law says, that the population is big, no mutation, and if at all it takes place it is in both the directions, those conditions are fulfilled, then the equation which the law gives says p² plus q² plus 2pq in a given population will always be 1. That means it will always be maintained as a constant. So what was the law saying? It was saying that the frequency, the relative frequencies of the alleles in a population, if the population is big, then it is constant from generation to generation. So this is what is called the Hardy-Weinberg Equilibrium. The factors that affect these or this equilibrium are 5 important factors. Number 1. That means the factors which would affect this equilibrium. First is mutation. And if we remember what we just now talked, we said that the law was or law is applicable when there are no mutations, and if at all there are mutations, they should be in both the directions. So if mutation takes place, then definitely it is going to affect the frequency of the allele. So this is one factor which will affect it. Second, recombination. Recombination takes place during gamete formation when the meiotic cell division takes place. At that time, if various kind of recombinants are formed, they can also affect the frequency. The third factor is gene migration. Gene migration is movement of some genes or alleles from a particular population. Here we take 2 terms, that is emigration and immigration. Emigration and immigration. Emigration means moving out of the alleles. So from here, if some alleles move out, then also the frequency would change. And immigration is when some alleles move into the existing population, then also the frequencies would change. So in this case, from the gene pool, some alleles move out, and in case of immigration, some alleles move in. If the migrations are in very, very small number, that means just few individuals are moving in or moving out, then it is not going to affect the population genetics. But if that moving in or moving out population is very, very different in its gene pool, then it will definitely affect the frequencies. The fourth factor is genetic drift. Genetic drift is by chance movement of allele. it can be elimination also. We have discussed this in detail, that if by chance some alleles are eliminated from a small population, and genetic drift is applicable when the population is small. Hardy-Weinberg's law is applicable when the population is big. So here they are talking about if the population is small, and from that population, if certain genes get eliminated by some kind of disease or some kind of accidental things, then that would be again affecting the population. One important thing which we can discuss here in genetic drift is that it works on the principle of founder effect also. So, genetic drift works on founder or founder's effect. This can also be understood from a bottleneck experiment. Suppose we talk of a bottle filled with colored pebbles. Say there are various kinds of pebbles in this bottle, and we know that the neck of this bottle is narrower, and suppose we put this bottle upside down. Some pebbles or some beads are going to come out. Say, and it is all by chance. Say only these beads come out when you invert this bottle. And say this is the population or individuals. When they come into one land or one position or one area, they would multiply, and when they multiply, they would form the individuals like them only. So the original 3, 4, 5 organisms which got separated, or in terms of genes, if few gene containing organisms get separated, they become the founder members of that population in a particular area. This is what is called the founder effect. Say from a population, some genes, they get eliminated, and those genes, they end up in some different area. The individuals having that particular kind of different gene, they become the founders. That means they will establish themselves in a particular area, then they will reproduce, and when they reproduce, the new individuals formed would have the same kind of genes as they had. So original individuals become the founder members, and this effect is known as founder's effect. That is also a part of genetic drift. And the fifth factor which affects this Hardy-Weinberg equilibrium is natural selection. The most important questions which have been asked on this particular topic are mainly on two things. One, on these factors, name or list the factors which affect, or they can give you various factors and ask which factor affects the Hardy-Weinberg principle. This is one type of question. And the second type of questions are numerical questions. That means using this particular expression, they would give us one kind of alleles frequency, and they would ask us to find out some other type of genetic makeups frequency. So let us take one example to understand how to solve questions which are based on this type of expression, that is on Hardy-Weinberg equilibrium. Let us solve one question to understand how this equilibrium works. Let us write down this one: p²+q² + 2pq is equal to 1. This is the Hardy-Weinberg equilibrium. Suppose there is a question that in Mn system of blood groups, the frequency of one allele, the frequency of M allele is say 0.7, and of the other allele, that is N, is equal to 0.3. Then what percentage or what would be the frequency of heterozygous individuals in the population? Of heterozygous individuals in the population. Now what is given to us is the dominant, that is, P, which we are representing by M here, that is given as 0.7. Q, that is recessive, represented by N here, is given as 0.3. And we are asked to find out the frequency of heterozygous. That means this is what we have to find out. So 2pq will be 2 into p square, into p, that is, and into q. So this is 2 into 0.7 into 0.3. If we calculate this, it should come to 0.42. This is going to be the frequency of the heterozygous individuals in a population. We want to just cross check it whether these frequencies are giving us this one or not. Let us write the same things here. P square would come to, that is, suppose, let me write it here. P square and Q square. P square would be 0.7 into 0.7, that is 0.49. Q square would be 0.3 into 0.3, that is 0.09. And 2pq we have already found out. So let us write it here. P square is 0.49, Q square is 0.09, and 2pq is 0.42. If we add this, the total is going to come to 1. That means we can cross check after solving the question. Normally the question is, they give us the frequency of one allele. Then we have to calculate the frequency of homozygous dominant, heterozygous dominant from that given value. We can also calculate the heterozygous and then place the value in this equation and cross check it by coming to that total of 1. So we said there are two important things on which questions are asked. One is this calculation type of questions, and second, the factors that affect the Hardy-Weinberg. So this is one very, very important topic of this particular